for the... Okay, thank you very much for the kind introduction. Yeah, maybe before I start, I would be interested who of the people in the audience has experience, besides Adriana, with small animal imaging? Just that I have a little bit of an idea. Okay, nobody. One. Fernando, yeah.
Yes, also me.
Okay, there are a few. Okay, at least. But most of you are probably from human PET or MRI imaging, so this is good to know. So I share my screen because this is for me kind of interesting. No, I'm not sure which one. Okay, and this is working, right? Let's see. Can you see my screen?
Yeah, we can. Yeah.
Okay. So just let me know if something doesn't work, then I will try to go back. Yeah, so I'm going to talk about how we map brain function and connectivity in rodents. And I'm especially working with PET and MRI to do that. And I will quickly introduce you a little bit to Tübingen, and maybe at some point in your life, you come to Germany, you definitely have to visit us here. Tübingen is a very small university town. Most of the people are probably students, or at least half of them. So during vacation time, it gets quite empty, but it's a beautiful city. So yeah, when you like to come to Tübingen, you have to visit also our labs. You see them here on the left side. So this is the imaging center, the entrance of the imaging center, and this is the radiopharmacy where the PET tracers are produced, basically. And this is what the labs look like. So we have to dress up completely to go into the labs, which is not always like super convenient, but at least we can make sure that we work in a very clean environment. So we have surgery labs, we have behavioral labs dedicated, with this red light for rodents. And then we have these IVC cages to have like a very clean environment, and these ventilated cages all the time. We have two MRIs, and one where we have the PET insert included. And then we have three, these were like the old scanners, but we have now 3 Mediso PET CTs. When it comes to small animal imaging, and that was the reason I was just asking about who of you has experience, there are a few things that may be an advantage compared to working with humans. One is that we can like highly standardize the groups in terms of, their genetic homogeneity. So we can have transgenic animals, we have their wild type litter mates, so we can keep those animals in a very controlled groups and in a very controlled environment, also with regards to food and so on. On the other hand, we have the ability to really selectively stimulate certain neuronal pathways by either using optogenetic tools, chemogenetic tools, or like other viral vectors to overexpress proteins, for example. And in addition to that, we can validate our results using ex vivo modalities or methods like immunohistochemistry, autoradiography, and so on, that we have like high resolution comparison to this data. And we don't have so much, the problem with the ethics approval for, radiation exposure. We don't need to recruit patients. So I think these are like the main advantages that we have when we work with small animals. There are for sure also a few disadvantages. And these are, first of all, that we always have to deal with the effects of anesthesia in our imaging studies. Then the small brain size is a problem. I mean, when it comes, from a mouse brain, it's about 2,000-fold smaller than a human brain. However, like the crystal size that determines the resolution of PET is maybe like 3-fold the difference. So this introduces quite a lot of challenges and in terms of partial volume effects and so on. Another limitation is for sure the complexity of human diseases. So we cannot model them exactly. And this kind, you can imagine, especially like in psychiatric disorders, depression, and so on. It's always like the question, can we really model the disease that we see in a human patient? So, and then, we need to think about the translatability. So whatever we find in rats is not always like completely translatable. In my lab, I'm focusing on doing imaging in terms of PET and MRI and the combination of the two to study brain function. We use different kind of stimulations, from optogenetic to pharmacological stimulations. We look at brain connectivity. We manipulate animals, using CRISPR, using optogenetics, chemogenetic tools. And then we also work on tracer development for different types of neurodegenerative disorders. And today I would like to focus on the PET and MR part, because this is also the topic here for the molecular connectivity working group. When we compare PET and MRI in terms of spatial resolution, and as you know, PET is more on the lower side. So with small animals, we are at about like 1.45 millimeters in the center field of view. However, we have a very high sensitivity in the nanomolar range, as most of you know. While for fMRI, we have the advantage that we have like very good time and spatial resolution. However, like the molecular sensitivity is rather low. So the insert that we work here in Tübingen with is still an insert that was developed. So this is the second prototype of an insert that was developed together with Bruker, but it was never commercially made available. Now I would say also a little bit unfortunate. But yeah, so the insert goes inside the MRI, and we acquire those data basically simultaneously during the scans. So if we have a neuronal activation in the brain, we can either look using BOLD fMRI on changes in blood flow, blood volume, or blood oxygenation. So the BOLD signal is a very convoluted signal of these measures. So we measure the hemodynamic response on one side with fMRI, and on the other side, we can look at these molecular changes with PET. This can either be neurotransmitter release, but it can also be like metabolic changes, as I will show in a few slides. A few years ago, there were the first papers published that kind of, that was kind of very interesting to use PET as a functional readout of in the brain again. So most of the time the tracer was injected as just a pure bolus, and then you could still look at which regions in the brain have more activity and less, but it was really difficult to do like these task-based stimulations that we know from fMRI studies. However, in these studies, they used a bolus plus constant infusion approach, and they could show basically that you could see this increased activation in the brain. And probably most of you know these studies that were published using this GLM approach. And what they showed in the paper, so this is the group from Vienna, that there was like a very good overlap between these activated brain regions that you see from the two modalities. And then this was further improved, or the approach was used using an independent component analysis also by the group from Shana Yamada in Australia. So they could also, using this data analysis approach, they could also show this nice overlap between what you measure with FDG and fPET or fMRI. So the question kind of comes always is, what do we actually measure in the brain? Is this like, if you have a stimulation in the brain, is this really FDG uptake from the neurons, or is this, the lactate that comes from astrocytes? And there have been 2 papers that have basically shown that during neuronal activity, there is an uptake, a direct uptake of glucose into these neurons to have like a very fast, metabolic response. So in the case of a neuronal activation, we see that glucose is taken up directly by the neurons. But it can also, and this may probably happen like more in a resting state, that you have also a lot of lactate transport from these astrocytes to the neurons. So it's probably both of it. But in any case, what we see in PET is the response that we see in PET is including both neuronal subtypes. So we cannot really distinguish neurons from astrocyte in the brain, but it would be a much faster process to use glucose directly from neuronal uptake. So the question is really, can PET, with the low temporal resolution that we have, can PET actually characterize brain activity beyond what we see in fMRI? So we set out in an experiment, and this was practically performed by Sabrina Buz in my lab, who really put a lot of effort in like establishing this fPET design and like doing the stimulations in the brain, together with Fernando, who's also here today, and who did a lot of the analysis in these studies. So the design was based on a fiber that we put in the substantia nigra. So this is a very well-known pathway, the nigrostriatal pathway. We used a loop coil for the stimulations. And then we put the animals on the scanner, and this was basically the stimulation design. So we had 10 minutes stimulation blocks, and then we always had like a 3-minute rest or baseline. And during these 10-minute stimulation blocks, we have always had like 60 seconds the laser on, where we had like the stimulation, and then 50 seconds off. So it was this on-off design. So 8 of these blocks within one of the 10-minute blocks. After the study, after we had also an anatomical scan, we took out the brains, we perfused them to make sure that we had a good viral vector expression, and then we did some more histological analysis. So this is basically the images that we found when we look at the T-map. So from the fMRI part of the study, so you see the strong hemodynamic activation in the striatum, so as we would expect it, mainly in the dorsal part, but basically in fMRI we see like very strong activation all over the striatum, and then some little parts in the midbrain. And here you see basically the bold responses in the different brain regions. So what was super interesting to see is that whenever we started the stimulation, we had a small increase in the bold signal, but after we stopped the stimulation, there was this overshoot in the bold signal that we found always in between these 15 second non-stimulations. And then we have, we didn't see that in the motor cortex or the S1, but basically we saw this in regions where we also have quite some dopamine release, so a little bit in the amygdala and the nucleus accumbens. In the substantia nigra, we found that there was like this increase but we didn't see any, like additional responses or decreases of the Wald signal during the resting periods. So when we look at FDG, I picked out like two important regions. One is the area where we stimulated the substantia nigra, where we had really the largest increase during stimulation, and then the striatum that also showed an increase over time. And when we basically plotted the signal changes compared to baseline, so we normalized the signal of this one stimulation block to the baseline signal, then you see that there's an increase of FDG over time, which was the largest, as shown on the left, in the substantia nigra, then in the right, striatum, thalamus, but there was not much, or like a slight decrease in the motor cortex as one. So these are the T-maps. We compared a few different methods because this was basically the first time that we did such a block design and an fPET study. So we wanted a little bit to see what method basically works best. So on the right side here in light blue, you see like this between sample approach where we compared channel-rhodopsin injected animals stimulated compared to GFP injected animals, which should not show any response. And here you see like the strongest response in the dorsal striatum and then the substantia nigra. And also like some areas here in the midbrain. But this was the most conservative approach because it's like a between group comparison. And then we had the ICA approach that I showed you, which was like a bit adjusted from what the Yamada group presented. And on the left side, the GLM approach from the Vienna group. So, and you see here you get probably strongest signal, which is also a little bit based on the fact that they take the response from fMRI into account. So they use like basically they normalize to a region where they know from fMRI that there's no response in the brain. So I would say the two approaches on the right are a bit more conservative. When we compare them directly, we could see that for both, we see the response in the striatum very well. It was a little bit stronger for both than for fPET. But what we found quite interesting that we had this very strong activation in the substantia nigra compared to board, where we only had like very few pixels that showed the activation compared to the PET signal, which was also shown when you look at the time signal changes, because the signal didn't show any changes between activation and rest. This is again the comparison. Here we see like the strong increase in the substantia nigra, a little bit, and then this overshoot that we always see in board fMRI. So we were kind of like wondering why, where does this come from? And the hypothesis here is that maybe, you have this very strong dopamine release. Dopamine goes to the autoreceptors in the brain and has like this auto inhibition, this like feedback loop, that it kind of like keeps these neurons non-activated, and once you stop the stimulation, that they go back to activation. So now we wanted to know, when we look at the fPET data, can we also use the time windows to look at connectivity changes? And first, I wanted to show from like a previous publication from Tudor Ionesco a few years ago already, where we did this comparison between fMRI and board. And here you see basically metabolic connectivity at resting state. And as you can see, the data don't really like completely correlate, which was not super surprising because we are looking at like two very different readouts in the brain. So we were interested, you know, like how does this look like if we have like this stimulation design? And so here again, we have the time activity curves from the PET. The problem here is that we have like quite short periods between these stimulations, but we could still find that this basically can be done when we use these 600-second stimulation time windows and 180-second baselines. And this was a study performed by Jan Ma. She's a PhD student in my lab, and she did on this data set together with Fernando also a connectivity analysis. And what you see here is basically if we put a seed in the substantia nigra, in the CPU, and in the right thalamus, this is the results for the connectivity that we get. So we see basically in the substantia nigra, we see connectivity between the nigra and the striatum. However, if we put the seed in the right CPU, this was like again like super interesting to see that this the CPU gets somehow functionally isolated there. So there was like connectivity within the CPU and with the nucleus accumbens, but there was no connectivity to other areas in the brain, which we found quite surprising. On the right side, we also used the thalamus to see, because the thalamus is somehow also connected in the basal ganglia loop, and you see there are some more regions that showed activity here. Now we wanted to see how does this look like for functional PET. And I think that was super nice to see that we actually see exactly the expected connections between the right substantia nigra that you see here down here and the dorsal striatum for putting the seed in the right nigra, CPU, and the thalamus, which kind of shows us that fPET in this case clearly shows the connectivity between the two regions if you do the stimulation of this pathway. So I would like to conclude a little bit that the advantages of using fPET for studying brain function connectivity is that we have like a direct measurement of glucose metabolism, and that shows us, you know, the neuronal activation of this pathway, that we get like a very stable signal over time. We don't have to deal with susceptibility artifacts that we often find in fMRI. We have also usually reduced sensitivity to motion or any vascular artifacts, and we can basically quantify using SUV or even kinetic modeling, which is easier in humans than in small animals, but it is possible to quantitatively access the stimulation. And then we have like a very complementary readout to what is used in board fMRI. There are however, as always, a few challenges that we have to deal with. One is that we have to fast the animals if possible before the scan. Otherwise, we have like a lot of FTG uptake already at baseline. We have to take care of anesthesia effects. We have a lower spatial and temporal resolution, which is for sure a limitation. And then we always have to deal with this cumulative glucose metabolism. Okay, now I'm coming maybe to a second study that I would like to quickly discuss, because it also kind of shows the advantage of combining the two modalities. And there has been like a few years ago this article in the New York Times about this psychedelic resolution. And as you know, like psychedelic have been in the focus since then, because they have been shown to have effects in PTSD, post-traumatic stress disorders, or depression, like psilocybin in depression and MDMA in PTSD. However, it's not really clear how these substances, work in the brain, what are the side effects. So we were just interested to use them in also a combined study and to compare what we see on molecular and functional level. So basically at baseline, we have a normal neuronal activity, and when we give MDMA, there's a blockage of several transporters, and MDMA has really the highest affinity to SERT over DAT and the norepinephrine transporter. So you have a blockage of the transporter and a high release of these neurotransmitters. So, and basically, since there's the highest affinity to the serotonin transporter, there's a lot of serotonin release. So this study was also performed by Tudor, Ionesco, and Mario Arment by that time. So what they did, they injected FDG as a bolus or a constant infusion. And these are the time activity curves that you see here. Then we used the 10 minutes before we started MDMA application as our baseline. And then you see really the increase of the FDG signal over time, which was lower in some regions and higher in other regions, as you can see here. So increase in the prefrontal cortex, several midbrain areas. In contrast to that, the board signal did not show this increase. So it basically had like a large decrease, which we found also like very surprising, that you have like these completely opposite signals from the two modalities. So the reason is that MDMA has a very strong vascular effect. So it goes also to 5-HT2A receptors, and they are expressed also on the vasculature. And this is the reason that we see like this vasoconstrictive effect and then the decrease in board signal. So when we do, they did the same study, also in combining carbon-11 dust, which is a PET tracer binding to the serotonin transporter. And here we saw the drop in the signal after the application of the MDMA, which shows us that there's this strong binding to the serotonin transporter as expected. And again, we could basically, in a separate group of animals, reproduce the decrease in the board signal. Since we were super surprised to see the changes in the board signal, so the drop in the board signal, we also put a region of interest outside of the brain, so just above the brain. And we could see that there's this overall signal drop in the extracerebral blood flow, which led us to conclude that this is not really a neuronal effect, but more like a vascular effect, which is always the problem we face whenever we do drug stimulations. So now, again, we were interested to see can we use these temporal fluctuations in PET, since we had this bolus plus constant infusion approach. Are these fluctuations meaningful? Can we use this again to look at molecular connectivity? And first, we did some tests where we looked at the animals that did not get an MDMA challenge. So we just wanted to see about the temporal stability of these measures using functional and molecular connectivity. So in the upper graph, these are the functional connectivity data at early time points on the lower left and at late time points on the upper right. And you see the correlation is very good. I mean, you see like a little decrease here on whole brain level, but overall it was a good correlation over time. And this was similar seen here for the PET data for the molecular connectivity. So here, this was a little bit lower with 0.8 correlation. Then when we look at the comparison, again, they don't look very similar. So that's why the correlation between the two modalities was not very high. And it shows us that we are basically looking at very different signals in the brain. We then performed an independent component analysis where we could see or identify 10 different components for the fMRI data, which was basically also previously shown in the literature, and 2 meaningful components for PET. So one was more in cortical areas or cortical midbrain areas, and one like in midbrain areas such as the raphe nuclei, where you have the projections from the serotonergic neurons to the rest of the brain. We then wanted to see if we we compared basically the changes in fMRI connectivity at 20 to 40 minutes, which is the baseline, and then at 60 to 80 minutes. Sorry, now I made a mistake. 20 to 40 minutes is not the baseline. It's basically right after the injection of MDMA. So it's early time points versus late time points after the injection. And then we had the two networks again, more in the deeper brain areas and in more the subcortical areas. And we saw in the fMRI data, when we use this sliding window approach, that there's a decrease over time, but it was not very strong. It was stronger for the so-called salience network compared to these serotonergic areas. When we now looked at the PET data, we also saw a decrease over time, which was, however, when we look at the change to baseline over time, much stronger, especially for areas of these of the raphe nuclei and these midbrain, deep brain areas. So maybe again, I would say that we have the advantage here that PET could pick up on a molecular level more signal changes or more changes in connectivity than what we could see with fMRI. And when we look at these two areas in the brain or in the regions that were affected, we could see that in these hindbrain areas, we had this immediate effect when we look at the time activity curves after MDMA challenge compared to the other areas that basically showed about a 10-minute delay in the response to MDMA. Yeah, so this again leads me to conclude that molecular PET can be used to study brain function. It's a very nice direct measurement of neurochemical processes in the brain. We have high sensitivity and specificity. It's non-invasive and allows us to do longitudinal experiments. And combination, I think it's a very complementary approach. I mean, for sure, we will not reach spatial and temporal resolution that we reach in fMRI. That's at least for now not possible. But maybe there we can work on approaches, to go down to maybe 10 seconds in the future. And I think it has many interesting clinical as well as research applications for the future. And with this, I would like to thank really all the collaborators, people that helped in these studies, funding agency, and my group that really has put a lot of efforts and is a really joy to work with on these experiments. So, and now I'm open for any questions. Thank you very much.
Great. Thanks very much, Christina. That was really interesting. Lots of exciting experiments with, preclinical PET MRI. So just goes to show that you can do a lot of things with small animals, not just in humans. So thank you for that. I don't know if there's any questions from the audience for Christina, that we could maybe start there. I have a few, but let's see if the audience has some to begin with. Anyone? Maybe I'll ask one while everyone is thinking about questions. So you showed a lot of interesting PET data with specific traces, FTG and then DASP, and so very targeted approaches. Have you considered looking at something that's a bit closer to the fMRI, so something like water or a perfusion tracer, to see how that compares, like the PET compares to the MRI?
I haven't done these studies. No, I mean, water is quite tricky and challenging because of the super short half-life. I mean, I know that a colleague of mine who left now for Siemens has done some of these studies. No, I mean, I was always, to be honest, mainly interested in animal models and not really like like I want to use the traces and models to look at pathology, to look at changes in, let's say, plasticity and neurotransmitter release. And I think this would more go in the direction of like maybe comparing, it's more like a methodology approach, but it would for sure be super interesting to do.
Yeah, absolutely. I mean, from the methodological point of view of kind of understanding a bit more about the bold signal, more than the PET part, because, with PET, absolutely, you want to look at specific targets, why would you go with a pan marker? But yeah, thank you. I think there's a question from Valentin. So do you want to go ahead and ask a question, please?
Thank you. Hi, Christina.
Hi.
Yeah, good. Thanks. Yeah, beautiful work. Thank you. Nice summary. So makes it easier to grasp the complexity. I had two remarks when I watched the slides. What was the first one? Yeah. Can you use your tracer, your viral tracer, to also shut off these dopaminergic neurons so that you can see whether you can diminish this sharp bold response? That would be just a methodological, like an interesting question from my side, for me.
Yeah, so this is possible. There are, instead of using channel rhodopsin, which like opens up the channels for neuronal activation, you can also have like chloride increase to kind of like shut them down. This was something actually I had in mind and we wanted to always do and test. But the first approach that we wanted to do, because what we did in the study is that we injected the virus in the substantia nigra, but not like completely selective to dopaminergic neurons. There's always, you might always hit like some surrounding neurons and the virus spread can be quite strong. I mean, you are more exact, with the fiber placement at the end, but still you will hit some surrounding neurons. So the first approach that we are trying now is to make this completely selective and use a Cree-rat model, to selectively stimulate these neurons. But this would be something we would anticipate in the future to also like do an inhibition of these neurons and see what happens.
Yeah, okay. Yeah, because as Adriana, I would have guessed that this sharp increase without the PET signal is then rather a neurovascular hemodynamic response. If you could test it, that would be great to see.
Yeah, I mean, there are also some of these PV neurons found in the substantia nigra pars reticulata. So this could also be somehow related to that. What I found, however, interesting is this, that we only found these increases in actually areas like the amygdala and the dorsal striatum accumbens, like dopamine innervated neurons. So we thought maybe, there's this auto inhibition because autoreceptors have like if you have this like strong increase in dopamine release, there's a activation of these autoreceptors. So that was something we would like to look into in the future a little bit more.
I mean, that's great, Christina, and a really good question, Valentin. That's one of the reasons why I ask about water, because many years ago in humans, water actually was used before fMRI for these sort of measurements, and they did saw some of these spikes. So interesting to see whether that's true as well in small animals.
Yeah, definitely.
Great. Mattia?
Hello. Hi, Christina. Thank you for
Hi.
your talk. Just an idea or maybe a stupid comment. Will TSPO PET tracers have any use of these functional PETs? Do you see any value of using a functional PET experiment with the TSPO or the tracer? Maybe to show TSPO, yes, the target for neuroinflammations. Would it show that it's functionally somehow manipulated by an inflammatory challenge, or would you not see any signal? I don't know if you're familiar with that tracer, so I was just curious.
Yeah, I mean, I've been always a bit careful with using TSPO. I mean, we have the GE 180 here in Tübingen. The problem with that tracer is that we don't have high brain uptake, at least like in normal wild type rats and mice, we don't see a good uptake into the brain. And I remember this recent paper where they discussed, I think it was in Nature Communications, where they discussed actually there's no TSPO on activated microglia in humans, at least. I think in animals, yes, but in humans, they couldn't find the expression, right? So I'm always a little bit careful with TSPO because it's so it's kind of contradictory in discussed in the literature. So no, I have not thought about it yet.
Okay, thank you.
And I think what I would find super interesting is these tracers, you know, like the SV2A, you know, these kind of that have like a broader uptake into the brain, that are not like selective for one receptor subtype. And I think I've seen already like some studies that have looked at this, or some other interesting like glutamatergic tracers. Yeah.
Thank you.
Yeah, that's a loaded question, Mattia, but especially with me as the chair of the session.
I know,
Because,
I serve it on a plate.
No. So for those of you who are not aware, we are working on TSPO in Edinburgh. So, and we are doing connectome analysis, but yeah, I think there's a lot to be said for TSPO. You're quite right on that paper, Christina. It's not that it doesn't exist in humans, it's the mechanisms in humans is different from the mouse, for example. So you have to be careful about interpreting the results.
Yeah, I thought they kind of made the point that it's on the activated microglia. It's not. Yeah.
It's basically a difference between having more TSPO and also having more microglia, or having just more TSPO, and that's the main difference between the mouse data and the human data.
Yeah. So yeah, maybe in this regard, if we say like more TSPO, more microglia could be an interesting.
And yeah, I mean, G180 is you're quite right, is not a trace that I would choose myself. There's just almost no brain uptake, which obviously if you have no signal, you can't measure much.
Yeah.
But yeah, I think we have another question from Xing.
Thank you, Christina, for the talk. I think the optogenetic part is very interesting, and also this suggests some kind of a directionality, right? You inject that region and then you see other regions also shows activity. I mean, try some kind of analysis on like causality. I think that is a very good model for causality, whether you have some like causal connectivity model. Have you done some analysis on that?
No, I have to say, no, not yet. I mean, as I said, Fernando, I think he's also here. I don't know. I don't see him. Yeah, he's here. So he has basically quite good experience in doing these type of analysis, but I have not. We have not tried it so far.
I think that is very good data to test causal models. And I have another like very naive question. And you said that you can use a very high level of radioactivity on the animals, right? How that can be compared with humans? How much a radioactivity level can that be for if you apply the FDG PET on humans? And so you will have a very good signals, right? But for human data, the signal.
I totally agree. This is I mean, what we did in the study was like our first attempt to use optogenetics with FDG. So we choose basically a protocol from the literature that had shown to have like highest dopamine release, because this is something, you know, we want to look also into at some point using fiber photometry, raclopride PET, for example. So we choose a protocol where we could make sure we have high dopamine release. This is probably not like in a physiological range. Let's say you would do any kind of task that you would do in an fMRI scanner in humans, like, you know, like visual stimulations or any kind of other stimulations. However, I think, I mean, the signal that we see in PET is so strong, and you know, like it's so specific. What we see, that I think we should be able, and I think with all these machine learning approaches and, you know, multivariate approaches, I think we can get a lot of data out from these, even in humans. So I would say this should even work in humans quite well. And I mean, yeah, at the end, you know, if we look in the human studies that I showed, you know, where they did this flicker board stimulation, it's also not like a very, yeah, it's also a very basic task, right? Like, I think more and more people are more moving into the field of naturalistic stimuli, and then we have to see how good we can resolve it, right? But I think the sensitivity of PET is high, and this is something we need to test in the future. Where are the limitations, right?
Thank you.
Great. Thanks very much, Christina. I think Valentin has another question for you.
Yeah, I forgot my first question before. Did you see in the fPET, you mentioned once that it was tricky that you had only a short rest period after the stimulation.
Mm-hmm.
But and on the slope, it looked that it not really had a plateau, but it was steadily increasing. But in your average response, it looked like it completely went back to baseline. So can you comment on that? So do you really see an fPET?
This is basically just the way how we analyze the data. So I only wanted to see a time window. You know, I defined a baseline time window, and then I looked at the first stimulation block and calculated the % change to baseline. And this stopped actually after, you know, when it went back to the next baseline, there was the analysis, the signal that it was not like a signal decrease in FDG. It was just like a change compared to baseline.
It didn't go back to the
no.
the
The
change,
FDG, no. It accumulates, right? No.
Right. Yeah. Yeah, no, I know it accumulates, but I wonder if your slope was going to 0 in the rest periods, or was it still increasing even in, like, it's always increasing, but
yeah. So during the rest period, it kind of like was stable. It didn't go up. Yeah.
Okay.
Yeah.
Great. Okay. One question from Ariana.
Hi, Christina. Thanks a lot for the talk. I really liked the initial slides where you tried to, well, gave us an outline of advantages and disadvantages of doing preclinical imaging. And one point was about anesthesia. So I was wondering, like, how much do you think that your results, that are quite encouraging for us PET people, would extend or apply also to normal wakefulness? And related to that, if you see any strategy to go around the need of anesthesia, like, for example, I know in some centers in humans, they actually inject neuromuscular blockers to keep the patient still, but perfectly awake. So removing the effects of the anesthetics. I don't know if in animals it's considered ethical to do, but
yeah, I think that's the biggest problem. I would love to do this, but we wouldn't get this through the ethics here. So they wouldn't allow us to do this because it would be much too stressful for the animal. I mean, there are some labs that do awake animal imaging, and I think if you have if you train these rats for a long period, so at least I would say a week or 2 or 3, you might be able to reduce the stress of the animal. You know, they get kind of like used to laying in the scanner. But you know, it's really hard. Like when we do the PET-MR studies, we can measure 1 animal per day. And, you know, having this like very long training periods and then doing this high throughput studies, and then you will lose a lot of animals because they move in the scanner probably, especially if you start stimulations. I mean, I have the experience whenever you give a drug, can be MDMA, can be amphetamine, can be optogenetic stimulations, you know, like due to dopamine release, they kind of go in a more wakeful state, and this can really like super challenging. And I know that it's always questionable if, you know, the anesthesia can affect the results. I mean, we also have them in the control, so I think there is an effect, but it might look quite different in non-anesthetized animals. That's for sure. Yeah, it's challenging. I don't know. I don't have the right answer. I'm always, you know, again, every year I'm thinking about if we should start this, if we should try it. But if you have like higher throughput studies, I think it's quite challenging. And what we need to think about when we combine the 2 modalities, the difficulty here is that for example, in PET, we usually use for the animals isoflurane, 1.5 to 2% isoflurane at high O2 concentrations, evaporated at 100% oxygen. This is an anesthesia that doesn't work for fMRI. You wouldn't see any bold signal. So we need to use an anesthesia, and there are these nice papers from Johannes Grandjean where they do these functional connectivity studies in rats. It's called the standard rat paper, where they use medetomidine with a low dose of isoflurane. However, and they show like it's closest maybe to the awake state. However, if we use medetomidine with FTG, we have like too high blood glucose levels, so it doesn't work, right? So at the end, we need to find an anesthesia that works for both, is not too confounding, and that's why we used alpha-chloralose in this regard. But yes, we cannot exclude that this may look different in an awake state, yeah.
Thanks a lot.
All right, well, great. Thanks very much, Christina, for a really amazing presentation, and thanks everyone for really interesting questions and discussion. I think we are really close to the hour, so we kept to time. I don't know if there's any other one last pressing question. If not, we can probably end today's session, and thank you again, Christina.
Thank you. Thanks for joining. And yeah, if anyone has like a question, just send me an email. I'm happy to chat about this. And maybe I see you at some conferences this year. Bye-bye.
Absolutely.
Don't forget
Sounds
to
good.
register to the next seminar. There's a link in the chat. Christine will be giving a very interesting talk as well. Thanks.
Great. Thanks,
Bye
Ariana.
bye, everyone. Bye bye.
Bye-bye.
Thanks, everyone.
Thank you,
Bye.
Christina. Thank you. Again. Bye.
Bye bye.
Bye bye. Thank you.
Thank you.